Hello and welcome. Dear students, today we are going to discuss an important topic, that is environmental effects of phenotypic expression and sex-linked inheritance. Let's first of all discuss environmental effects on phenotypic expressions. Phenotype is the observable physical or biochemical characteristics of an individual organism determined by both genetical makeup and the environmental influences. For example, height, weight and skin color. The concept that phenotype represents the consequence of genotype-environment interactions, that is GEI, is universal and relates to all living organisms. Gerard was one of the first scientists to note that the effects of genes on phenotype could be modified by the environment, that's E. He suggested that differences in the genetic composition play a major role in the variable response to the drugs, and that this effect of the genotype G could be further modified by diet. Similarly, Teresen demonstrated that the development of a plant is often influenced by its surroundings. He articulated the existence of a close relationship between varieties of crop plants and their environment, and stressed that the presence of a particular variety in a given locality is not just the chance occurrence, rather there is a genetic compound that helps the individual adapt to that area. He pointed out the role of selection in driving the genotypical differentiation of the population in a given locality. Wright further elaborated the existence of a functional relationship between various biological endpoints and networks of genes and environmental factors in his studies of mutation, selection and breeding. In humans, environmental effects are complex and their relative importance may vary considerably according to the genetic constitution of the individuals. The physical environment can include such factors as pollutants. Physical, social and economical factors further conspire to derive GEI through complex environmental insults such as tobacco, smoke. As a result, GEI can influence disease onset, the rate of disease progression and the clinical response to pharmacological intervention. The rapidly increased prevalence of many common human disease traits clearly illustrates the importance of GEI on disease onset. Asthma rates in the USA have risen dramatically over the past 20 years. Since this is too short a period of time for most allele frequencies to change significantly, non-genetical factors, that is change in the environment, must be important. Similarly, the doubling of obesity rates seen in many countries during the past few decades did not result from change in the underlying genetic architecture, but rather from alterations in the environment, primarily what people eat and how much they move. The human genome initially evolved to handle exhausting physical activity in lifestyle where resources were scarce and famines were common. For example, traditional hunter-gatherers lifestyle. As we move to a modern lifestyle, our genome has not had time to adapt sedentary living with abundant food and material goods. Thus, a large percentage of the population in USA and the other developed countries is at risk of developing complex disease including obesity, diabetes and hypertension. Just as the genome is made up of genes controlling the phenotype, the environmental consists of an amalgamation of biological and physical factors which independently or combination affect the genome. If we wish to predict how a living organism will develop, we must first know genetic constitution that it inherits from its parents. Thus, we must know the historical sequence of environments to which the developing organism is exposed. Every organism has a developmental history from birth to death. What an organism will become in the next moment depends critically both on the environment that it encounters during the moment on its present state. It makes a difference to an organism not only what environments it encounters, but in what sequence it encounters them. A fruit fly, that is Drosophila, for example, develops normally at 20 degrees Celsius. If the temperature is briefly raised to 37 degrees Celsius early in its pupal stage of development, the adult fly will be missing part of the normal vein pattern on its wings. Dear students, now moving on to another objective, that is sex-linked inheritance. In dealing with sex-linked inheritance, we are mainly concerned with those genes which are carried by sex chromosomes or allosomes. It may be assumed that each of the sex chromosome, X and Y chromosome, consists of two segments where one of the segment remains homologous between the two chromosomes, X and Y, so they are called partially sex-linked. Such traits are sometimes transmitted as simply as autosomal traits, either through X chromosome or Y chromosome. On the other hand, the non-homologous segments of X and Y chromosomes are of great significance because true sex-linked genes are located on them. Completely Y-linked genes are called holondric, and this type inheritance is extremely rare. In some cases, they are considered doubtful. Also, rather the genes on the non-homologous segment of the X chromosome are the common type of sex-linked genes, and the term sex-linked is usually referred as X-linked traits. Since these genes are mainly carried by X chromosomes, they are commonly called sex-linked genes, and the peculiar pattern of inheritance followed by them is termed as sex-linked inheritance. Dear students, moving on to the next part, that is sex linkage in Drosophila. In 1910, when T.H. Morgan was working on the genetics of the fruit fly Drosophila langoster, in his laboratory at Columbia University, he came across a male fly having white eye color. In Drosophila, the normal eye color is red. The white color arose due to mutation. Morgan searched for the pattern of inheritance and discovered the presence of sex-linked gene. There are 4 pairs of chromosomes in Drosophila. Of these, a pair of heteromorphic chromosome is concerned with sex determination. The male is heterogametic X and Y, and the female is homogametic XX. The X chromosome contains a large number of genes and mainly consists of euchromatin. The Y chromosome is largely inert and has no alleles of any genes. As such, it is composed of heterochromatin. Therefore, in the male fly X and Y, the phenotype is determined by the genes present in the X chromosome. For the presence of a single X chromosome, male fly can be termed as hemizygous. This means that the male possesses only one copy of genes in the place of normal 2. In contrast to this, the female fly carries 2 sets of second-linked genes in their 2 X chromosomes, X and X. Since there are 2 sex-linked genes, alleles, what we call, in the female fly, the dominant gene expresses itself. If both the genes are recessive genes get the scope of express themselves. But the situation is different in male flies because he by nature possesses either a dormant or a recessive gene, and in either case the gene expresses itself. Therefore, all male flies are capable to show the trait, whether dormant or recessive. The characteristic feature of X-linked inheritance is that here no transmission of traits is possible from male to male, that is, father to son, because X chromosome of the male, that's father, is not transmitted to his son, rather it passes to daughters. But the same father can transmit his sex-linked traits to grandsons through his daughters. Therefore, the phenomena has been referred as crisscross inheritance in genetics. The crisscross pattern of inheritance is possible only when the mother is homozygous recessive, that is XX, and the father bears the dormant XY gene. Dear students, moving on to the next part, that is sex linkage in men. The genes present on the X chromosome of the man follows the same pattern of inheritance as in the sex-linked traits of Drosophila. A close examination of X and Y chromosomes in the man reveals that the majority of the genes for the X-linked traits are found on the portion of X chromosome, for which there is no corresponding homologous region in the Y chromosome. The Y chromosome is comparatively small size and contains very few genes. Moreover, just like Drosophila, man is hemizygous. He has only one X chromosome. However, the characteristic features of inheritance for a sex-linked trait can be summarized as follows. Number first, ordinary genes governing sex-linked traits are not transmitted from male parents directly to their male progeny. Number second is, a male transmits his sex-linked genes to all his daughters, since females receive one of their two X chromosomes from father. These daughters in turn transmit this gene to half of their male progeny. Thus, a sex-linked recessive gene is transmitted from a male to its female progeny and thereafter to half of the male progeny of such females. Number third is, since sex-linked genes are not located in the Y chromosome, the heterogametic sex, that is human male, is hemizygous for such genes, that is, it possesses only one allele of sex-linked genes. Therefore, recessive alleles of sex-linked genes express themselves in the hemizygous conditions, while they have to present in homozygous state in order to express themselves in the homogametic sex. For example, in human, the long arm of Y chromosome is homologous to the short arm of X chromosome. Genes located in that region of X chromosome does not show sex-linked inheritance. Some well-known examples of sex-linked human traits are hemophilia, that's inability of blood to clot on exposure to air, color blindness, that is inability to perceive one or the other colors. For example, green color, that's optic atrophy, that is degeneration of the optic nerve. Genuine glaucoma, that is hardening of eyeball, and myopia, that is nearsightedness. Defective iris, epidermal cystis, and white occipital look of hair, mitral stenosis are some of the examples of these. The situation can be explained in this way. If a male with any of the above mentioned defects marry a normal homozygous female, all their sons will be normal, but their daughters will be the carriers, that is heterozygous, for the particular disease or defect, although they look almost normal. This condition is produced due to the sex-linked recessive gene. When the daughters, that are the carriers actually, get married to normal males, 50% of their sons will be perfectly normal, while remaining 50% will get the defect. A few concrete instances will help in understanding the situation. The best known examples of sex-linked in man is the color blindness and hemophilia in man. Now, let us discuss red-green color blindness in man. Perhaps the best known example of sex-linked in man is the color blindness. Several types of color blindness are known, but the most common type is red-green blindness, which is an X-linked recessive trait. Persons suffering from this color blindness cannot differentiate between the red color and the green color. Individually, the red color blindness is referred as protanopia, whereas the green color blindness is known as deuteranopia. The genotype for normal vision may be symbolized by XX and the color blindness by XX'. X indicates the sex-linked recessive gene for the color blindness. Now, let us discuss hemophilia in the man. Another sex-linked recessive gene in man is hemophilia or bleeding disease. In this disease, blood fails to clot easily on exposure to air, so that even minor injuries may cause prolonged bleeding leading to death. A woman carrying a gene for hemophilia in the heterozygous condition, that is XX, does not exhibit any visible effect of the disease, but is capable of transmitting the disease to 50% of her sons. Hemophilia is very rare in women because to have this disease, the woman must be monozygous, that is XX for recessive genes. Among the children of the hemophilic man, all the daughters are carriers of hemophilia, but none of his sons are affected by the disease, provided the mother of the children is neither hemophilic nor a heterozygous carrier. Hence, there is no scope of transmission of X-linked traits from father to son because of crisscross pattern of inheritance. With this, we come to the end of today's program. Hope you have enjoyed it well. Thank you for watching and goodbye.